Hello, everyone. My name is Joseph Bellissimo, and along with Tianyu Liu and Reynald de Leon, I'm extremely proud to welcome you all to iGEM Toronto's 2020 PET project, Enzyme Engineering for PET-based Textile Degradation. Slide. The COVID-19 pandemic and global shutdown was particularly effective in emphasizing the threat and widespread nature of plastic pollution. Slide. Plastic production is increasing, and current recycling solutions cannot keep pace with less than 10% of plastics being recycled. To make the issue worse, plastic production and the rate of plastic production is increasing with a 100 million ton increase in textile plastic production within the last 10 years. Slide. Even the gold standard for recycled plastics, polyethylene terephthalate, sees only 1 out of every 5 tons produced being recycled. Slide. Current popular recycling methods, such as mechanical recycling, result in lower quality with repeated cycles and can't deal with contamination, while chemical recycling is expensive and energy intensive. Thus, the cost of virgin PET is much lower than that of recycled PET, which further drives demand away from recycling. Slide.
So, plastic waste is a huge issue that cannot be solved with a single solution. So our stakeholder research revolved around specifying our focus. Some minor technical difficulties. I'm sorry, there's a lot. I think my laptop is not able to handle it. Sorry about that. It should be working now. Okay. So, we initially thought that enzymatic recycling would be best used for PET bottles. But multiple stakeholders advised us to look to address issues with no particular solution. We learned that plastic bottles were efficient enough, or mechanical recycling was efficient enough for plastic bottles. And coupling this with the reoccurring notion that fabric recycling has no solution, we decided to explore the avenue of textile waste, in particular synthetic textile waste. Now, polyester is created from PET and makes up approximately half of total textile waste in mass. The overwhelming majority will end up either being thrown out into landfills or being incinerated. This is because conventional methods, aforementioned mechanical and chemical, do not work for polyester. And with Canada unable to ship off waste anymore, we'll need a solution for increasing amounts of textile waste here. So we learned that collecting and sorting is difficult for general plastic recycling. But through our stakeholder research, we were given suggestions into looking at donation centers, which offer us a feedstock. And particular clothing items within those donation centers, in our case being the jersey, could be used for our proof of concept, which we'll explain later on. sorry, I'm deviating a little bit, but perhaps it would be more efficient to just play it from this instead of the slideshow. So sorry about that. Now this slide is all numbers, but what it essentially calculating is the rate at which the enzyme would need to operate in order to degrade PET to produce it at the market price. And that rate is 36 times faster than the fastest one developed thus far, the Eternia enzyme.
Nonetheless, a biological solution can maintain the properties of the recycled plastic and is resistant to contamination and impurities, making it a sustainable long-term answer to the plastic problem. The biological depolymerization of PET is facilitated by hydrolase enzymes, particularly PETase and MHETase, which were discovered in 2016 and shown to convert PET to its monomers ethylene glycol and terephthalic acid. Here we can see the structures. Here we can see the structures of both MHETase and PETase. MHETase is composed of a lid domain, which is designed to specifically recognize the MET substrate, and a hydrolase domain, which catalyzes it into monomers. PETase is composed of only a hydrolase domain with a very wide active site cleft, which facilitates the entry and subsequent hydrolysis of up to 4 subunits of the very large PET polymer. Recently, a novel protein engineering attempt was made on the enzyme LCC, generating a variant with the highest PET depolymerization rate to date. Unfortunately, this enzyme is still significantly too slow at depolymerizing PET to be industrially competitive. Thus, we employed a novel protein rational design approach on the PETase, MHETase, and LCC enzymes in order to enhance their catalytic activity and thermostability to that required for cost-effective industrial integration. Our novel rational design pipeline facilitates the production of multi-mutant enzyme variants with diverse analysis across the entirety of the protein, without having to rely on flawed energy functions. The result of this approach and further analysis generated the following top-scoring variants. Our Toronto PETase variant had 43 mutations from the wild-type PETase, and a Rosetta energy score that is 20 kilojoules per mole lower, while still maintaining a very similar backbone structure. Click. Our Toronto LCC variant comprises 32 mutations from the initial Tournier LCC variant, which is the best PET depolymerizing enzyme to date. Click. Our Toronto MHETase variant contains 57 mutations from wild-type MHETase and has a whopping 200 kilojoule per mole lower energy. While our novel rational design approach did generate very interesting variants, its tools are limited to relying heavily on ancestral and existing sequences. Thus, we need tools like machine learning to explore areas of the sequence space that are very different from our existing enzymes.
To get a sense of how large the protein sequence space is, you can compare the number of possible sequences for our protein, which has length 263, to some other fairly large numbers. Here, if you were to imagine the scenario where every single atom in the universe were proposing 10 to the 16 sequences per second, and all these atoms have been running in parallel since the beginning of time, you can only explore a tiny sliver of the possible sequence space for our enzyme. So clearly we need efficient machine learning algorithms to explore the space. Right, so here's our machine learning pipeline. We start with a generative model that can generate protein sequences, and also a discriminative model that can tell how good a protein sequence is. That's in that yellow box on the left. And then during generation time, we just generate new protein sequences and use that oracle that can predict how well our generated sequences are to score the generated sequences that's shown in the box in the blue. Finally, we filter these sequences based on their score, and we retrain the generative model on the sequences are predicted to perform well. We iterate this a couple times, and we eventually get the sequences. Next slide. So there's motivation from last year where we used the same method to design to generate the sequence. And next slide. Even though it has a pretty low sequence identity, you could see that it did show some promising activity that was indeed better than the wild type. Here, the ML variant is shown in that gray line there. Next slide. So this year, with some tweaks that you can see the details of in our wiki and in our full-length video, we are able to control the sequence generation process more precisely. And here is one of our generated sequences. Next slide. Right, so on the wiki, you can see a video of our MD simulation, which essentially simulates the interaction of the protein with the ligand, the plastic ligand. So here is just a well-type PETase, and the distance, that graph you see on the right, it tracks the distance between two important atoms participating in the reaction. The closer they are, the more likely the reaction it is to occur. Next slide. So we can validate the wet lab's designs. So what we did was we did MD simulation on their design, their best design. And here you can see that interatomic distance is very low. Here you can see in the plot it's in red, and it's pretty stable throughout the simulation. So this is an idea that can tell us, well, this enzyme is able to stably bind to its target ligand, much more stable than it does to the wild type binds to the ligand. Next slide.
So with our newly constructed protein variants complete, we have designed both an iterative and integrative hypothetical laboratory procedure. The iterative procedure will make use of in vitro p-nitrophenol butyrate assays to empirically determine our enzyme variant's catalytic activity and differential scanning calorimetry to determine its melting temperature. Our integrative pipeline makes use of lambda-red recombinering, which allows for the efficient and stable integration of our enzyme variants into neutral loci, such as LAC-Z, in the MG1655 genome for heterologous expression. Next slide. So now that we have assembled a bacteria that is capable of secreting these plastic-degrading proteins, we can throw them in a bioreactor. During cultivation, pressure, temperature, and pH are the three key conditions of the reaction. The product mixture, including our target product, EG and TPA, unreacted PET, E. coli cells, enzymes, LB nutrients, buffers, and other byproducts should be fed to the downstream purification process to isolate and purify the EG and TPA. The purification process uses phenol to dissolve PET, followed by another filtration to isolate the undissolved TPA. Additionally, after centrifugation to separate the suspended E. coli and protein from the liquid solution, a distillation column can be performed to separate EG from furfural based on their different boiling points. Next slide. So here we show the results of our cultivation simulations using Optimus Firm. So it predicts that complete substrate degradation will take place in 10 hours in the top right graph, and it also shows expected levels of products in the lower graphs. Next slide.
So what should all this theoretically look like put together? Well, we'd start at the top with our feedstock, we'd collect it from donation centers, and then we'd bring them to a processing facility where we would sort, decontaminate, and turn them into particles the bioreactor can use most efficiently. After enzymatic degradation, we would prep the desired byproducts, terephthalic acid and ethylene glycol, which can then be sold to make cosmetics or antifreeze, and even back into PET. Now, as for our outreach this year, it consisted of two main avenues, outreach events and our social media. Our events consisted of educating individuals already interested in synthetic biology about current ethical issues, as well as giving them a panel of experts. Our social media involved engagement, and broadcasting iGEM's events throughout the summer.
So in summary, we have designed a novel method of rational design that doesn't rely on flawed energy functions, a novel machine learning approach for generalizable de novo design of optimized proteins, and a set of enzyme variants along with their genetic architecture and a full bioreactor process with the potential to be used in large-scale industrial PET degradation. Next slide. So our future directions include validation of our enzyme variants using our iterative procedure and its associated assays, testing the generalizability of our machine learning approach and its ability to optimize other diverse proteins, and finally to integrate our best variants into our MG1655 microbe and bioreactor using our integrative pipeline to test their efficiency in PET depolymerization. Next slide. So thank you all for listening and coming to view our summary presentation on our work this year. Thanks again.
Yeah, so thank you for the presentation. Right now we're just opening up for the questions from the judges. So we're just gonna have about 20 minutes and then open to the general. So judges, if you have any questions, you can ask them now. Or type them in the chat if you'd like.
So first off, great work. I'm quite impressed. So I had a couple of questions. So you mentioned that your design approach doesn't depend on flawed energy functions. How do you simulate energy functions?
Okay, so the thinking is essentially that there are energy functions that are taken into account, but their biggest flaws are minimized essentially. So if we talk about the rational design process, one of the things we do is at the very least to consider a mutation beneficial to the protein when we generate a large number of them. It can't just be the fact that it decreases the Rosetta energy units. It has to be at least by 3 Rosetta energy units, for example, which significantly decreases the chance of false positives. Another big thing that we use is we don't make decisions and don't rank variants just based on the Rosetta energy function, which is a very common thing that people do in rational design. Instead, there are many other factors taken into account, such as, especially in multi-mutant enzyme variants, where there's a lot of epistatic effects that can happen when you combine mutations. What we are able to do is actually we're able to design an algorithm to see which mutations kind of go together well. So we see what pairs of mutations associate with each other, and even though those may technically have a lower Rosetta energy function, we can use them together. There's additionally a bunch of other structural features in in some of the programs that we use that eliminate our need for Rosetta energy function. So for example, there's a few mutations that structural predictions suggested would make huge impacts to our backbone structure, but were favorable via the Rosetta energy function, so we chose to ignore those mutations. So essentially our design process doesn't rely on it and doesn't make decisions based on it.
So here's what I don't understand about your approach. Is your approach trying to enhance the affinity to the ligand, and that's your output that you're trying to optimize? Or are you trying to optimize catalytic activity? Because those are two separate things. It could bind the ligand very well and just retain it there without doing anything to it, right?
Yeah. So essentially we kind of divide our work into kind of 2 different parts. There's some most of our mutations are actually not even close to the active site. And those are just ones that mostly like just increase the stability of our protein. So we use something called PROS mainly for these types of mutations. And they increase, while maintaining the backbone structure, they might increase like hydrogen bonding or hydrophobic interaction. So a lot of these types of mutations. Now, we use a very specific program called FuncLib when we look directly at the active site, so the first contact shell. And in this case, FuncLib is, you could, essentially, it's, how it works is it stabilizes, it identifies ways of stabilizing the ligand, and you can also input information about kind of the catalytic process, so the transition states. It is able to make it's able to make like very limited predictions about the chemical process. So how it works is essentially it's trying to stabilize the interactions with the ligand and bring them relatively closer, which is associated with increased catalytic activity, but not necessarily causes increased catalytic activity. So it's a certain methodology, but it doesn't necessarily always work.
So at the end of this entire design process, candidate that you want to test or a variety of candidates?
We have hundreds of candidates. Yeah, we have hundreds of candidates, but we use a ranking method and we showed our best variant. But we have hundreds of candidates to potentially test.
And how did you prepare for the MD simulation? Did you do any relaxation, any of that preliminary stuff beforehand, or did you just run it off of the X-ray crystallography? What did you do?
Yeah, so Rosetta Relax, we needed to relax the designed structure with all the mutations. And so one really important thing is to have a good starting conformation for the MD, especially its interaction with the ligand. So we did multiple rounds of docking with Autodock Vina, and we filtered based on the conformations that had the most stable binding energy between the ligand and the design. That's really important to have a stable MD simulation. Otherwise, the results are not going to be meaningful.
There is no structure known of this design, before activity, during catalysis, and after. You don't have like a before, during, and after snapshot that's available on the PDB.
Right. So you mean like the transition state of that reaction? Yeah. So that's something we're looking into. It's difficult to model those structures in the PDB. I'm not aware of any crystal structures that.
So this is purely modeling work. You're not even starting with a foundation that's built off of crystallography, right?
No. So the crystal structure of the protein exists, but that's the wild type protein. And when we add mutations to it, we just relax it, minimize the energy with Rosetta Relax. As for the transition state of the protein and the ligand, that we don't have. It's very difficult to model that conformation. But essentially what all the other papers have done in these MD simulations is try to model the stability of the interaction of the protein with the ligand before the reaction. And that, as the 28L paper showed, can be a useful proxy of determining the catalytic activity.
Mohammed, are you finished with your questions? Okay, thanks. Great presentation. I just have a few questions. I think in your presentation that's posted on the website, you had mentioned for one of your mutations, and correct me if I'm wrong, that it's most efficient at pH 9. Is that correct?
Is that, yeah, potentially. I think the optimal pH for our enzyme is around pH 9, yes.
Okay, and I was just wondering, like, how feasible is it to maintain that pH for E. coli to remain functional if you're using that to produce it?
Yeah, so that's a great question. Like in the bioreactor, I'm assuming you're referring to, is that right? Yeah. Yes. So there has been precedence of other research teams working with their microbes at pH 9. they have not used MG1655 directly. So it may not work. However, I believe that testing directly in the bioreactor will give us a better sense of the feasibility of this. And it should also be mentioned that the enzyme still shows quite strong activity even at pHs ranging from 6 to 9. So 9 is the optimal pH, but it definitely is highly functional. Like it doesn't go to like 50 or 10% at the lower pHs. So it's still very highly functional from pHs of 6 to 9.
Okay, thank you. And then my next question that was more experimentally based too. In your experimental design, you mentioned using the lobster cell strain. And I was wondering, since it is meant to be more efficient for nickel affinity purification, if there's the potential for any extra contaminants that might come with this, if that might interfere with your enzymes capabilities.
Yeah, so the lobster strain is pretty much the only use of that chasis is kind of to isolate obviously and purify our enzymes via the hexahistidine tag. Since our data from last year really kind of suggests that it's unlikely to have any effects on our purified proteins when we do the in vitro assays. So our work from last year kind of strongly confirms that there is no background in the lobster strain that would impair the function and mess up our approximations of the catalytic activity.
And then I was just wondering about like your end process, where you show like how your system would work in general. I was wondering if this integrates well into infrastructure that's already present in these textile processing buildings and facilities, or would they have to like have a cost to them?
So thank you for the question. There will be associated costs because right now large-scale textile recycling doesn't exist, at least for synthetic textiles. So we would either need to sort of retrofit an existing organic textile recycling plant to process the polyester, or we would have to invest in that processing equipment in the beginning.
Last question. It's just more if you guys could comment, because I don't know if I missed it in your other video. But if you could comment on how your collaborations have worked throughout this year for like within your team, with other teams, and then if you had any additional research labs who you've been in contact with.
Well, I can comment a bit on intra team or inter team. I'm not sure which one. So yeah, there's actually quite a great linkage between all of our work, especially because the HP, especially between HP and their economic analysis and business analysis, wet labs rational design, kind of the two-pronged wet lab rational design and dry lab machine learning. So we have MD simulations, which are constantly filtering back between the wet lab and dry lab. And we have those economic analysis of not only our pathways, but also our bioreactor processes and simulations, which connects us very well to the HP economics side. And a lot of the decisions we made were informed kind of by interviews, like why are we targeting textiles in the first place? It's because a lot of people have shown that this is the real issue in the plastic production that mechanical and chemical fail to address. And I think it really makes sense that HP kind of really informs the design work by dry lab and wet lab, which is constantly cross-talking with simulations and comparison of data and comparing our structures. And so, yeah, it's a very beautiful, I think, approach that meshes us. So in terms of collaborations with other teams, so first of all, we didn't have really access to our laboratory, which is kind of a big negative. we weren't able to directly collaborate with any other teams, just because I don't think many other Canadian universities were able to access their laboratories either. So everyone's kind of doing their own design work. So yeah, that's kind of really held us back, unfortunately.
Thank you.
No problem.
So if there's any more judges with questions, or if any of the audience would like to have any other questions, you can either type it in chat or you can turn on your audio and ask here.
So while they think of other questions, so you're culturing these pre-processed textiles with in the bioreactor with these enzymes, or with the bacteria that's producing the enzymes. How are you then, after that, isolating the byproducts that you're going to be selling? Hello?
Can you guys hear him, just to make sure?
Can you guys hear me?
Yep.
Okay. Do I need to repeat the question?
Yeah, I didn't hear the question.
Okay.
I'm not sure if anyone else.
So you mentioned that you're pre-processing these textiles, breaking them down so that you can, I think it was either culture them with the bacteria that are producing the enzymes, or just with the enzymes themselves. But then your major, how you're making money off of this, is you're taking the byproduct after breaking down these textiles and you're selling those to cosmetic companies and whatnot. How are you isolating the money maker from the reaction process?
Yeah, so yeah, that's a great question. I think that's well explained in our kind of isolation. We have, in our video, we have a whole explanation of how we isolate ethylene glycol and terephthalic acid in the bioreactor section. To keep it short, essentially PET and TPA are put in one kind of stream, and we're able to isolate them by dissolving PET with phenol. And the other stream is EG along with LB and buffer, sorry. And so we treat that with LLE and furfural as a solvent, and then use distillation because there's different boiling points, and we're able to isolate EG. So there's a whole section in the bioreactor that kind of talks about how we isolate these specific products from our depolymerization process. So yeah.
Awesome.
I guess I have another question too. I was just wondering if, since you guys have done like a lot of like really great MD research at this point, do you guys know how many mutants you're going to be testing experimentally and trying to validate their activity now?
Jenny, do you want to talk about the what is it called? The loop pipeline that we set up?
Oh yeah. So this year, yeah. So with all these designs from this year, the idea is to filter. Obviously, we can't test hundreds in the lab. So the plan is to filter these by their predicted score and their energies, and perhaps synthesize 5 to 10 in the first batch, just to make sure that the purification process is working, the proteins actually fold, right? But after this first step, next year, what we do plan on doing is to really tightly integrate the ML side with the wet lab, using an active learning approach, where, depending on how much money we have, that's really our limit. We can order a batch of sequences every month or so. And from that experimental data, that can be fed back into the machine learning model that can predict catalytic activity and thermal stability for each of the designs. And we envision this would happen about 3 times over the summer next year. And this would generate if we get maybe let's say 20 or so sequences per batch, and that would be done for 3 to 4 rounds.
Like how many amino acids?
So PETase. Oh, sorry. You go.
Oh yeah, sorry. PETase is 263 amino acids without the signal sequence.
Just 27.
Yeah, LCC is about the same, but MHETase is around 600.
Very big. Okay, thank you.
Thanks.
If we had, if anyone from the audience would like to have any questions or say something, you can type it in chat. Give like a few minutes, but then just to let you guys know, 3:15, the session ends, and then at 3:30 there's also another workshop. So, any other questions you guys can ask now too? Next 2 minutes. If not, you can always, there's also the Slack after you guys are done. You can always chat with everyone.
Sounds good.
So, can you guys hear me?
Yeah.
Yes.
Not a question, but yeah, I'm impressed with the quality of work you guys are doing. Great job. If any of the people from iGEM Toronto is going to pursue grad school, consider the Michael Garten Lab. We do mammalian sim bio work, and yeah, I think you might enjoy it.
Thanks so much. We really
Thanks for
appreciate
that,
that. That means a lot.
Yeah, he's no, he does protein design.
Yeah, exactly.
It's pretty
So
cool.
it's right up the alley of this work. So
Yeah, it's been a lot of fun. I think COVID kind of derailed a lot of initial plans, but it really allowed us to enrich our work in protein engineering, and we found that as such a rewarding and really cool field to work on, and it's very tightly linked to synthetic biology. So that was very exciting.
yep, that's why I'm in it.
Very good skill to have.
Awesome. Looks like we don't have any more questions. So maybe if judges can regroup after everyone else is left, that would be good.
That's great.
Sure.
Thank you for the presentation. I should be better on a Zoom call because this is still being recorded.
I'll send the Zoom link.
Great. Thank you, everyone.
Thank you.